Okay, so we're going to take a step back from the social sciences and the social component now and delve a bit into ecology. And I'm going to talk a little bit about the impacts of global change on lionfish and the lion fishery. And I just acknowledge that I've got a huge amount of co-authors that is on the work that I'm about to present, and we've had a huge amount of institutions that have supported us over the time that we've been doing the research that we've done. To give you a bit about myself, as mentioned, I'm at Rhodes University. My research focus is really on all aspects of coastal fisheries, including governance, human and ecological dimensions. I have a particular interest in climate change impacts and then the complex interplay between these dimensions. But for this talk, it's really going to be a little bit about the climate change impacts on the ecological dimension of our coastal fisheries. And to just give a quick summary, I'll first introduce the lion fishery and how complex it is. And I'll look at the environmental changes that are likely to impact lionfish. How will temperature impact our lionfish populations? How will changes in pH impact our lionfish populations? And then how does exploitation and climate change, how do they interact with one another, and what are the potential consequences? And then finally, I want to end off with some ideas of how to future-proof our lionfish populations and our lion fishery. So to give you a little bit of an overview of the lion fishery, it's probably, in terms of participation, well, it is the biggest sector in our coastal fisheries. It's broken up into several subsectors: a commercial boat, small-scale boat, recreational boat, small-scale shore and recreational shore sector, each with a large number of participants. We don't have a load of information about the catch, with the exception of the commercial boat-based fishery. And we also don't have a large number of information of the jobs associated with these sectors, although the recreational fishery supports an enormous amount of jobs in coastal cities and towns. But overall, if you look at the participation and the jobs that we do know about, we can see that the lion fishery is clearly critical for livelihoods in our coastal environments. And the lion fishery, if you look at it from a systems perspective, as we've heard a lot about that in the last couple of talks, but is incredibly complex. And it has a societal system that ranges from everything from the poorest of the poor that are literally eking out just fish for food, to super wealthy recreational anglers that literally spend millions of rands on going fishing. And so it's a complex social system. And then the ecosystem is equally complex, because each of those little subsectors actually compete for similar resources. And if not managed carefully, there's really room for unjust sort of distribution of resources. This ecosystem is then, and so the societal system is obviously run by a governance system, which is supported, which is basically led by the Marine Living Resources Act, as well as the small-scale fisheries policy. And in terms of climate change is obviously going to have big impacts in terms of the societal system, so on people, but I'm really going to focus on how it's going to impact the ecosystem. And I'm going to try to make the linkage of how we can use the science of climate change to inform best governance practices. So that's really what I want to be talking about today. In terms of our coastal lionfish, there are a large number of environmental or climate impacts that are likely to have an influence on them. The first, obviously, is the sea temperature and its gradual and increasingly rapid rise over time. And while mean temperatures do certainly have an impact on fishes, it's that climate variability, that it's ocean weather, that tends to be the thing that really tips these fish populations over the edge. So if you look at the top right, you will see just a little graphic of some of the thermal range observed between March and May in 2018 in Titicaca and Port Elizabeth. And you can see the enormous range of temperatures that these fishes literally have to endure. And when these little peaks or drops actually, slip past the tolerance of a coastal species, we run into serious trouble. So it's these real weather events that are really the things that are more sinister than a gradual increase in temperature. With regards to the other factors, there's the changing pH. We're having a reductions in pH, and that is increasingly or exponentially reducing over time. And this will have an impact on our coastal fishes. And then we have the development of low oxygen zones all over the planet. And in South Africa, that's mostly driven by things like eutrophication and red tides and upwelling potentially. And these all have impacts on our coastal fishes. And the kinds of impacts that they have are, they influence the demography and the distribution of our fishes. For example, in terms of demography, it will influence the growth of our coastal fishes. And then distribution is self-explanatory. We've already seen several instances where coastal species have shifted long distances poleward in response to ocean warming events. And then they also influence the phenology and behavior of things. In other words, the phenology is the timing that things happen. And an example of that would be reproductive timing. And you can see an example of that would be the spawning seasons of some fish might be getting shorter and shorter. And this may have major impacts for recruitment. And in terms of migration, the timing and of the migration patterns might be changing, and this could have significant consequences, because when the fish arrive at their site for either feeding or breeding, the appropriate resources or environmental conditions will not be there to fulfill those components of their life history. And all of these things together, therefore, have an impact and a consequence and makes fish species ecologically vulnerable. And this is directly passed on to fishes. And it basically translates to socio-economic vulnerability. So understanding the ecological component and the ecological vulnerability is absolutely fundamental if you want to understand what kind of social vulnerability we're going to see in order for us to make adaptation plans to try and figure out how we're going to manage the rapidly changing conditions. So in 2015, we did a review of coastal, our expected changes of coastal within the coastal fishes and fisheries. And we made some predictions on what is likely to occur. And this graphic basically illustrates most of the predictions that we made. And we separated the fish out by their life histories, such as migratory species were different from residential species. Migratory species would, for example, rapidly shift their distribution following their preferred thermal regime. And it was largely a poleward distributional shift because the east coast and the upper west coasts tend to be warming rapidly. The east coast is also, the current strength is becoming greater, the Agulhas current. And we're likely to see the migratory species shift south. The patterns were less clear in the south because there's no clear signal of thermal change. And that's because we're seeing increasing warm events from the Agulhas current, and those are punctuated with cold events from increased upwelling events that we see. And so that, of course, has major impacts on the resident species. But the resident species, the type of impact won't be as quick necessarily as a migratory species. It will just shift, but it will over time alter its growth patterns because it won't move, but it will remain stationary, and conditions will likely become unfavorable over time, and it will decrease its reproductive scope or the potential for reproduction. And so slowly for the resident species, we'll begin seeing these similar changes happening as you'll see with the migratory species, but the actual mechanisms are a little bit different. So this summary, this review was based on a review of existing literature and the expected environmental changes that we're seeing. and then the likely responses of fish to these, but they were not based on knowledge of the mechanisms driving these changes, and also didn't consider the combined impacts with exploitation. And this has really been the focus of our research since then. So I'm going to try and share some of that with you. So if you want to understand the impacts of environmental change, you need to look at environmental limits, and this is really the crudest method of understanding whether fish will be able to survive in future conditions. And that's simply as using temperature as an example. You basically put a fish in a tank and you basically heat it or cool it until it loses equilibrium, and that's considered its upper and lower limit. It's called CT min and CT max. And then you look at the environmental conditions and you have a look to see whether those are substantially different from those thermal tolerance points. And the difference between those is known as your thermal safety margin. And what we found for a range of species is that actually our fish fell well within, they had broad thermal safety margins and largely could tolerate the temperatures, certainly now and probably way quite a bit into the future. We then looked at some other factors, and included in that was respiration, so breathing rate. And we had a look at where there was a break point, when suddenly at the temperature where there was a change in the rate that fish were respiring. And we found that there were significant breakpoints, which indicated to us that there was some kind of physiological mechanism going on at certain temperatures. And what we did find was when we looked at those breakpoints, if you could see in the bottom figure here at the BP, you can see those breakpoints actually, there was very limited thermal safety margins, certainly at cooler temperatures for one of the species that we had a look at, suggesting that these cooler temperatures might have really important sublethal effects for our fishes and might have large impacts down the line. We then wanted to understand whether fish could actually acclimate their CT min and CT max temperatures. And so we acclimated fish at different temperatures, cooler and warmer temperatures. And we found, interestingly, that if you subjected them to an acclimation for 170 hours of a warmer temperature, their CT min and CT max levels actually increased. And that was quite a positive finding, well, partially. If you think along the east coast of South Africa, where the conditions are warming, it may indicate that there might be some potential for adaptation or for acclimation over time. However, when you look at the south coast, which is increasingly subjected to marine heat waves and marine cold spells, if you could picture a marine heat wave followed, which would have a would be equal to a warm acclimation temperature, followed by a marine cold spell, those fish would definitely not be able to survive those temperatures. So this variability that we're seeing along the south coast is a real concern for us in terms of the survival of coastal fishes. And what I will say is we're increasingly seeing these large upwelling related fish kills along the south coast of South Africa. And it might be related to the prevalence of heat waves followed by these upwelling events. But ultimately, if you want to understand the response of fishes, looking at the endpoints are only one piece of information. The other is really rooted in their response to different environmental conditions and throughout the continuum of that they're likely to be subjected to. And the only way that, well, one of the ways that you can look at the impact of a particular environmental parameter on the performance of a fish is through physiology research. And so you would find, for example, if you have a look at environmental parameters, say, for example, temperature, you would see that fish perform poorly at a certain range of temperatures, then at a suboptimal level, and then at an optimal level. And increasingly, I think we're going to be seeing our fish subjected to suboptimal and poor conditions. It doesn't mean that they're not going to survive, but it means that they're not going to be as efficient in the way they do things. For example, their growth and reproduction might be impacted. And to understand that, we need to do a great deal of physiological research. And we are incredibly fortunate to have the SciAps ecophysiology research platform, which has given us literally world-class infrastructure to do the kinds of research that we need to do to understand how the physiology of fish will respond to different environmental stresses. And the great thing about this lab is that we can work at all life stages, because literally the responses of an individual species will change per life stage. And so understanding the whole, whether a population or a species will be impacted, requires understanding of each particular life stage. But we can actually do that, and we can measure the metabolic performance of these fishes at the different life stages in this facility. So it's an incredible investment that SciAp has made to get these kinds of research facilities available to us. And so if you measure the physiological response of a representative number of fish at each life stage in a population, you can actually begin to understand the potential population response. And I'll give you an example of it. We took some of the physiology equipment up to Namibia, and we looked at two congeneric species that are very important in their coastal fisheries. They're cob species. One is a warm species, Agarizomus corodus, and the other one is a cool water species, Agarizomus inodorus. And we looked at their metabolic physiology and their aerobic scope, which is AS, which is the right-hand figure, which is essentially the amount of energy that they have for things other than keeping alive. And we found that there was a clear change in the performance at warmer temperatures, and Agarizomus coronus became dominant at the warmest temperatures, indicating that as the environment warms, which is predicted for the region, they are going to become the species that dominates the fishery. So that would be how you would convert this into some kind of understanding of the potential changes that you're going to be seeing. Other techniques that we've used are using heart rate loggers. So we basically insert a heart rate logger using surgical processes into the fish. We change the water temperature in the lab, and then we monitor the heart rate. And you can clearly see that there's a linear increase in heart rate up until a point, which is a break point, which suggests that there's some suboptimal impact happening on these fishes where they are no longer performing at their optimal level. So it's a way of understanding where those subtle changes are that you might not see ordinarily. And then taking those kinds of pieces of information, that physiological information, how do you actually apply it and turn it into something that is? Well, the answer to that is using mechanistic species distribution models. So what we do is we take that information, we develop a model of the distribution of the species based on what we know about its physiology, and then we predict forward based on future conditions. And what we see in this figure is a current contemporary distribution, or a 2005 to 2009 distribution of red Roman, where that environments would be suitable for them. And then you can predict to a future distribution, which I think was 2100. And you can see some changes in the potential distribution, particularly along the wild coast. There seems to be a large reduction in the area suitable for these species. And that's so using those physiological metrics are incorporated into the species distribution models. But we would like to take these a step further, because naturally this particular species and many others are resident reef fishes, and they can only literally inhabit places that are suitable for them. So if we could bring some geosciences into this, we could use seafloor mapping to understand where the available habitat for these reef fishes is, and we can really tweak these models to indicate where we are likely to find these fishes in the future, and then we can start creating adaptation plans for things like the line fishery. Fish growth is also likely to change over time, and I mentioned that earlier, and one of the ways that we look at the potential drivers and potential changes in fish growth is we use dendrochronology. So we look at the otoliths of the fishes, and the otolith is basically provides a growth history, like rings of a tree. And what you can do is you can measure the width of the increments in the otolith. So if you have a look at the otolith here, you can see the distance between these dots is essentially the width, and it it is related to the growth of the fish. So where the dots are really broad, such at the beginning of their life, they're growing faster, and then right at the end they grow a lot slower. But the relative distance of these dots gives us an idea, and we can date it based on from when the fish is captured back, so we can give it a date. You can then, with a number of these outlets, look at what the growth was like for the species in a particular year. And if you can get that information, you can then match it with environmental parameters, and then you can start understanding what kind of environmental parameters are driving growth in the species, and then you can predict forward based on what you know is going to happen in the inshore, in the coastal environment. In this example, the autumn temperature was highly significant in terms of its predicting what the growth rate for this particular species would be. So ultimately, you can use this information to estimate growth rates of your species without having to do the research, and then incorporate this into stock assessment, which then inform the regulations for the line fishery. Other things that you can do is in terms of looking at fish reproduction, and we know that reproduction is going to change. An example of some of the work that we did there was we looked at the range of temperatures in which a species is known to reproduce, and then we plotted over time what has happened and potentially what is likely to happen in the future in terms of their reproductive performance. So if you look at the right-hand side here, you can see, for example, in Namibia, which is in Angola, sorry, the white dots, and Namibia, the sea bream is likely to reduce in its reproductive potential. So there's a negative change in the months that are suitable for spawning over the year, whereas in Namibia, it's likely to increase. So that's suggesting to us that this species is going to decline over time in Namibia, but increase in time in Angola, but increase over time in Namibia. And we can use this information to predict where these species are likely to do well, who's going to be the winners and who's going to be the losers in terms of the coastal fish populations. Now, in terms of ocean pH, there was quite a lot of work done, and we did quite a lot of work, and more recently there was a review that looked at a meta-analysis review that looked at the impacts of pH on fishes, and largely what they found was adult fishes tend to be quite resilient to the impacts of ocean acidification. They have very good acid-base regulatory systems, and it doesn't seem to be incredibly energetically expensive to adjust your pH, and they do it through their gill tissue. But there was less clarity on the early life stages, and so we focused in quite a bit on the early life stages and how ocean acidification could affect those fish. And one of the experiments that we did was we looked at dusky cob, which is a very commonly caught species and are personally important in the aquaculture industry. We had a look at the metabolic scope of these early life stage, so literally from egg through to flexion stage, which is like before they're even juveniles, you won't even recognize them as cob. And we had a look at what their metabolic physiology looked like, and we found that at 15 days, which roughly coincided with when these fish began to develop gills, they suddenly stopped performing well in the low pH conditions. And what we also found, interestingly, that more or less at the same time, there was a big decrease in both ossification and chondrification. In other words, the formation of the skeletons of these fishes, which for us indicated that there was probably an energetic bottleneck, firstly, but secondly, that there might have been difficulties in actually assimilating calcium, which is some of the building blocks of bone tissue at the early life stages. And this is a real concern for us because there might be significant developmental challenges and early mortality for our coastal fishes in a future ocean. But I do want you to just remember this, because I want to present a study a little bit later on that will actually show that there is maybe hope for these fishes. Okay, so that's talking a little bit about the impact. So we can clearly see the major impacts from climate change on the fishes themselves. And then you have the fishery, which in turn is having an impact. And we wanted to know what the combined impact of these things would be. And so what we did know about angling is that, and in the line fishery, people fish with hook and line. They selectively remove certain individuals for the fish populations, and it's selective. For example, they remove bold individuals, in other words, individuals that are likely to take a bait from a population. And the consequence of that is that the population becomes more timid. They also remove individuals with faster growth rates, which means ultimately the population growth will slow. And ultimately, they reduce the diversity within the populations. But there are links between the growth and the physiology of fishes, and also between boldness and physiology of fishes. So therefore, by that train of thought, angling should also remove certain metabolic physiological phenotypes, and that could really result in reduced physiological diversity and potentially the adaptive potential of these fishes. And so when you're thinking about a fish population, we generally think about it as a population, and we draw a thermal performance curve, for example, where we know that their performance will be poor, suboptimal, or optimal. But within these populations, there's a huge amount of diversity. And we found that in one of the PhD students in my lab, Lauren Bailey, she had a look, and while there is a population curve, you can see an enormous amount of diversity, which suggests that the raw materials for adaptation is there. And we just need to make sure that we don't remove it. And we wanted to ask the question, is hook and line fishing actually taking away some of those raw materials? Is it taking away some of that diversity? And how does it affect the physiology of these fish populations? So we went into the lab and we took some highly resident fish, which stayed in a very defined area, red Roman, and we tested their thermal physiology, and we compared a long-standing MPA Titicaca with a heavily exploited area, similar environmental conditions. And then we compared another highly exploited area of Cape St. Francis with Chokama marine protected area. And we found in both of these studies that the aerobic performance, so the physiological performance of these fish, was better in the marine protected areas, without a shadow of a doubt, and particularly at the thermal extremes. And then we thought, well, this is a lab study. Does it actually translate to wild behavior? And are we actually seeing differences in behavior in these different environments? So we tagged fish in the same sites with acoustic tags. We set up a very fine scale array, and we stuck accelerometry tags, which actually measure the acceleration of the fish over like a highly variable upwelling period. And we found that the patterns that we saw, acceleration and activity of the fish in the marine protected areas, was always higher than in the marine protected areas, and particularly at the thermal extremes. And so our lab results actually mirrored the wild results. And we found that fishing was really actually, well, removing those important, well, having an impact on the physiology. When we looked at the individuals in that telemetry experiment, we found that there were fewer individuals that maintained their activity at extreme temperatures in the populations that were exploited. And that suggested to us that angling actually removes individuals that have a broad thermal range of aerobic performance. In other words, those ones that are going to be good in the future, we're taking out. But then we also wanted to know whether boldness and physiology were related, and did boldness play a role? And so one of my students had a look at a link between physiology and behavior of fishes. And she first categorized individuals as either high performers, intermediate performers, or low physiological performers. Then she linked that up with behavior, and she did activity, boldness, and aggression tests on the different individuals. And she found that without a shadow of a doubt, every single individual that had a high physiological performance were also more active, more bold, and more aggressive, suggesting that these were the first ones that were likely to be removed from a population through hook and line exploitation. So we then finally understood the mechanisms that were operating on this thing. So essentially, in an unexploited population, you'll have high thermal performance phenotypes and low thermal performance phenotypes, which you can see on the left. Over time, these high thermal performance phenotypes feed at all temperatures, in extreme conditions, optimal conditions, and so extreme low and extreme high conditions, and they are selectively removed. They are also selectively removed more so than the low thermal performance types at optimal temperatures. So eventually what you end up with is low thermal performance individuals in your populations. And this was a fundamental thing that we needed to understand. But what that potentially means is it means that fishing is making fish less likely to survive climate change. And they're probably less adaptive if the genes of these low thermal performers are then passed on to the next generation. So we wanted to test that. So we got a PhD student to go out into the lab and have a look to see whether individuals spawned from a marine protected area actually were different from individuals that were spawned in the exploited area. So we literally field spawned these fish, we took them back to the lab and we subjected them to different stresses. We chose not to do a thermal stress experiment because of the very narrow range of temperatures that these early larval fish can tolerate, and we didn't want to kill them all immediately. So we chose pH. So we had the exploited offspring and the protected offspring, and we subjected them to a pH stressor. And we found that after around 5 days, there was a significant metabolic depression on the fish from the exploited population. These recovered later, but we felt that they only recovered because they were in a highly food-rich environment. We basically had so much food in those tanks they could feed all the time. In the natural environment, I doubt that this will ever be the case. And we felt that this metabolic depression was at the early life stage was a real signal that the offspring that are born from exploited fish populations are no longer as resilient as those born from fish in protected populations. So what it says to us is that fish populations generally have the raw material for adaptation, but exploitation is literally inhibiting their potential to adapt. And so that brings me on to some of the final parts. So how do we fix this? What do we do to make sure that our fish are adaptive, they can acclimate, and they can survive a rapidly changing climate? And the first thing that we need to do is we need to maintain diversity within our fish populations, and physiological diversity particularly. And so one way to do that another way to do it is to promote the likely phenotypes that are going to perform in future oceans. So for example, along the east coast of South Africa, which is warming rapidly, you want to try and protect the individuals that will perform at the highest temperatures. Whereas along the South Cape coast, which is fluctuating crazily with marine heat waves and cold spells, you want those individuals that are both thermally, that can do well at those thermal extremes. So like the fish on the right-hand side. So how can we get to that space? Well, the first thing is clearly from our results, we need a strong network of sanctuary areas around the coast. And these are the ones that are going to protect our fish and maintain that physiological diversity. And of course, what they will do is at spawning time, the eggs and larvae will move outside of the marine protected areas and seed the areas and maintain the fisheries that we desperately need to maintain over time. So the next step is to plan where these MPAs need to be to ensure that the fisheries that they're currently supporting are protected. In other words, find where the fish are coming from and make sure that those fish are protected. Okay, the second thing that we can potentially do is we can restrict our fishing activities at thermal extremes. This is going to be much more of a challenge. But I've already got a competitive recreational angling group to agree to shifting their fishing competitions when we have extreme weather events, so really warm conditions, for example. And so that might be one way that we could try to protect those future performance phenotypes. And then the last way is potentially considering genetic rescue, and that will literally be taking adult fishes potentially from protected areas and actually planting them in exploited areas, and hopefully hoping that they're going to seed the surrounding areas with their kinds, with these high physiological performance phenotypes. So that is a rapid whirlwind of some of the things that we've been thinking about and mulling over for the last sort of 8 or so years. And I will say that the kind of work that we would be doing would not be possible without the support and the infrastructure that we have around us. The SciABS Aquatic Ecophysiology Research Platform is just an incredible place for research for us. And then all the work is facilitated by sandparks, Cape Nature, etc. And this has really helped us do the work that we can, and hopefully we can make a contribution down the line.